Welcome back to my lecture videos. Dear learners, as you can see, I will be discussing on the industrial production of glutamic acid. So you can see the different approaches for production of the glutamic acids are direct chemical synthesis, fermentation, bioconversion using different types of enzymes. Out of this, we are going to discuss about fermentation process in this lecture video. So let's proceed into the details of the topic. Glutamic acid, what are the different properties? I have given a brief description about this particular compound. The chemical name is L-glutamic acid. Molecular formula is C5H9NO4. Molecular weight is 147.13 gram per mole. Color is usually colorless to white. It is usually odorless. Solubility is sparingly soluble in water and insoluble in ethanol and ether. pH is between 3 and 3.5, and this is the molecular structure of glutamic acid. If we go for the historical perspective, we will see that the first glutamic production dates back to around 1908 when Dr. K. Ikeda, a chemist in Japan, isolated glutamic acid from kelp and discovered that glutamic acid, after neutralization with caustic soda, developed an entirely new taste. In 1908, Kikuine Ikeda of Tokyo Imperial University isolated the flavor enhancing principle from the seawood of konbu as a crystals of monosodium glutamate, popularly known as MSG. The breakthrough in the production of MSG was the isolation of a bacterium named as Corynebacterium glutamicum. The successful commercialization of monosodium glutamate with this bacterium provided a big boost for amino acid production. Glutamic acid commercially production by microbial fermentation provides 90% of world's total demand, and remaining 10% is met through the chemical methods. For the actual fermentation, the microbial strains are grown in fermenter. The raw materials used include the carbohydrate, peptone, inorganic salts and biotin, that is the composition of the inoculating media. Biotin concentration in the fermentation medium has a significant influence on the yield of glutamic acid. Fermentation completes within 2 to 4 days, and at the end of the fermentation, the broth contains glutamic acid in the form of its ammonium salt. So you can see that 90% production relies on the process of fermentation. They must overproduce glutamate in excess of their normal metabolic needs. They must excrete into the culture broth. The most popular coryneiform species includes Brevibacterium flavum and Brevibacterium lactofermentum. Other glutamic acid producing organisms include Escherichia, Megatherium, Bacillus circulans, Bacillus cereus and Sarcina lutea. This is the flowchart form representation for the production of glutamic acid by fermentation process. First of all, you take the nutrients, dissolve it in the media and take it in the dissolving tank. Then you sterilize the media by different processes such as autoclaving, radiation. Then you take it to the buffer tank to maintain the pH. Then you put it in the fermenter. Inoculum of the strain is put it here, and other pH control and sterile air is also included. Then the cell separator acts after the process of fermentation is complete. The cell separators separate the mass, solid and liquid. Ion exchanger is acting over here, and we are taking the ammonia for reuse. The liquid product can be eluted with sodium hydroxide, and then it is undergoing ion exchanger therapy. We go for the evaporation process and crystallization is done, and the L-glutamic acid or the monosodium glutamate is obtained in form of pure crystals. This is the media composition and conditions. What are the different components and their percentage in composition? The media must contain glucose with 8%, NH4Cl, that is ammonium chloride, 0.5%, corn steep liquor 0.3%, dipotassium hydrogen phosphate 0.5%, magnesium sulfate 0.03%, calcium carbonate 1%, and deionized water to make up the volume. The pH of the medium is normally kept at 7, and the fermentation is usually initiated with continuous agitation and aeration for around 48 hours at 30 degrees centigrade. So, here you get the pH, the time, incubation time, and also the temperature. Next, we come up with the process of separation and purification. In this particular step, we are going to utilize 4 types of process such as centrifugation, filtration, ion exchange, and electrodialysis. Centrifugation is a common method used in industry, can be operated with semi-continuous or continuous basis. Poor centrifugation can be improved by adding the different types of flocculating agent. This agent will neutralize the anionic charges on the surface of the microbial cells. Filtration is widely used in industry and can be improved by using the different filtrates. This separates unnecessary cell debris and impurities. Ion exchange is used for the extraction and purification of amino acids from the fermentation broth on the basis of charge of the solution. Electrodialysis is based on the principle that charged particles move towards the electrodes in the electric field. A mixture of the required amino acid and contaminant salts can be separated at the isoelectric point. The applications are limited to desalting amino acid solutions. Here you can see the diagrammatic representation of the two processes named as ion exchange chromatography and the electrodialysis. Ion exchange is based on the separation on the base of positively and negatively charged ion. Positively charged protein binds to negatively charged bead and the negatively charged protein flows through. So, this is how they can be separated. Here also electrodialysis method is there. In this, on the basis of charge, the ions are separated. Okay. Separation and purification. Solvent extraction has only limited application. The distribution coefficients of amino acid between organic solvent and water phase are generally small. Decolorization is performed to get rid of the colored impurities in the fermentation broth. It is based on the fact that amino acids do not adsorb onto the activated charcoal. Although the treatment is very effective, some of the amino acid is lost during this step. After the glutamic acid crystal cake is separated from the acidified fermentation broth, it is added to the sodium hydroxide solution and converted into monosodium glutamate, which has a strong umami taste. The monosodium glutamate is cleaned by using active carbon, and the clean monosodium glutamate solution is concentrated by heating. The monosodium glutamate crystals is formed. The crystal produced are dried with hot air in a closed system. Then the crystals are packed and ready for marketing. What are the different applications of glutamic acid? They are used in nutritional supplement in food production. The monosodium glutamate is used as a flavor enhancer. It is also used in agricultural or animal feed, and it is used as an intermediate in manufacturing of various organic chemicals. This MSG, that is the monosodium glutamate, is a very famous flavor enhancer. It is thought to have some amount of carcinogenic effect if it is overused in any type of food. So, I have covered every aspects of the fermentative production of glutamic acid. I conclude my presentation here. Thank you for following my lecturing videos. If you have any queries, you can drop into the comment section of my YouTube channel, and my students can drop into the queries in the Google Classroom platform or personally mail me.